we're going to talk about some technical tips for qPCR assay design and setup. I'm just going to give you a basic overall view of what I'm going to cover today. My main focus is going to be qPCR hydrolysis probes. So I'm going to start with the basics of qPCR. We will then discuss some difference in capabilities between SYBR and 5' hydrolysis qPCR. I will go into more detail into primer and probe design criteria for 5' hydrolysis assays, and finally give you all some suggestions and ideas on setting up experimental experiments. So why is qPCR better than traditional endpoint PCR? Traditional endpoint PCR makes measurements at the end of a reaction when all reactions have reached plateau phase. Consequently, you are unable to measure differences between the starting amount of your sample amongst your different samples of your target DNA. Whereas with qPCR, you're making measurements during the exponential phase and during each PCR cycle. This gives you the ability to quantitate differences in the starting amount of target DNA between your samples. This consequently leads to decreased variability in your results, as well as the ability to have a high throughput in the number of samples that you can analyze. How is real-time PCR different from traditional PCR? Real-time PCR has the same components as regular PCR. You have the buffer, the water to provide the matrix, buffer to maintain your pH and salt conditions, your template target DNA, primers, DNTPs, which are the building blocks, Taq polymerase enzyme that synthesizes new target DNA, and magnesium chloride, which is important for primer and DNA stabilization, as well as for optimal activity of your Taq enzyme. So in addition to these regular PCR components, qPCR has an additional method of detection. There is the addition of a fluorescent probe, and because of this fluorescent probe, it makes quantitative estimation possible. This fluorescent probe can be either in the form of SYBR green, which is a fluorescent intercalating dye, or a dual-labeled hydrolysis probe, or molecular beacons, or FRET probes, or scorpion probes. But as I said earlier, my focus is going to be on the dual-labeled hydrolysis probe. So these probes most commonly have a 5' fluorescent dye attached on the 5' end, such as FAM, and a quencher that is attached on the 3' end. At IDT, we have a new and improved version of the dual-labeled hydrolysis probe. We have an internal quencher that is known as the double-quenched probe. The quencher is Zen. This provides even additional quenching capacity for this probe. So the idea being is that as long as this probe is intact, any fluorescent energy emanating from the dye is captured by the quencher. However, when this probe degrades, it separates the fluorescent dye from the quencher, and consequently the quencher is no longer able to capture that energy. This fluorescent energy is then detected by the instrument and recorded for every PCR cycle. Before I go further, I just want to clarify, there are a lot of names for dual-labeled hydrolysis probe-based qPCR assays. In the literature, they're called 5' nuclease assays, which is the same thing as 5' hydrolysis assays, also known as TaqMan assays. At IDT, we call them the IDT PrimeTime assays. In terms of basic mechanical mechanism of action, they all work the same. So going on to the mechanism of action for qPCR 5' hydrolysis assay, it's the same as in a traditional PCR assay. There'd be 3 phases. The first phase where you denature your DNA at 95 degrees and you separate your 2 strands of DNA. Next, the temperature is lowered to allow for annealing of your primers and probe. It's at this temperature also that extension also begins to occur. Some cycles also have a third phase at 72 because that is when the enzyme can be optimally active. Regardless of the temperature, either 62 or 72, extension will occur from the 3' end of the primer. And as it extends, the polymerase then comes across the probe that is hybridized. There is one unique feature about the Taq polymerase. It has a 5' to 3' exonuclease activity. So this capability makes it possible for the polymerase to then digest this probe, which is in its way as it's synthesizing DNA. This digestion, in effect, separates the fluorescent dye from the quencher. So this separation, whatever energy was possible to be absorbed by the quencher, now because of the separation of the dye and the quencher, this energy can no longer be absorbed, and this dye starts fluorescing. It's this fluorescence then that is captured by the instrument. So the polymerase would go on to synthesize its complementary strand of DNA. So this would what happened in the first cycle, and then in consequent cycles, more and more target DNA is amplified. As more target DNA is amplified, more fluorescence is detected by the instrument. So fluorescence release is measured for each PCR cycle, and the fluorescence that is released is directly proportional to how much target DNA is synthesized and amplified. In comparing SYBR versus 5' hydrolysis assays, SYBR is inexpensive, so it's a very commonly used dye. However, keep in mind that non-specific PCR products and primer dimers can also contribute to obtaining a fluorescent signal. So as a consequence, this use of SYBR also means that you have to do post-analysis melting point determinations to make sure that your signal is only due to a single amplicon and not due to multiple amplicons. Another disadvantage of SYBR is that you cannot do multiplexing. You cannot analyze multiple genes at the same time, or you cannot do genotyping with the standard SYBR. 5' hydrolysis assays have increased specificity over SYBR just because of the fact that there's a third sequence in the assay, the probe, that gives it, that makes it more specific. It's also useful when you're trying to amplify rare transcripts or you're trying to detect pathogens, and it's also advantageous because there's no need to run post-run melt curves. The main attraction for 5' hydrolysis assays is the ability to be able to use multiple dye quencher combinations for multiplexing, as well as you can perform SNP genotyping. 5' hydrolysis probes can be slightly more expensive. We at IDT, however, have offered the PrimeTime Mini for our customers for the discovery aspect of genes, so that you don't buy more reactions that you need, and it's less expensive than buying a whole lot of reactions. So what goes into a successful qPCR experiment? First and foremost, you have to start with excellent primer and probe design to make sure they have the right TM, the right sequence complexity in terms of location and secondary structure. Following that, you want to make sure that you have the right number of biological replicates, the controls that are needed, and so that the data that you obtain is valid. Often overlooked are these areas of RNA DNA isolation, purification and quantification, and reverse transcription. These areas also can drastically influence your qPCR data. Finally, of course, your qPCR reaction setup in terms of the technical replicates that you have and the controls, as well as the master mix used is important. And after you've done all this, you have data to analyze. Due to time constraints for this talk, I'm going to only be able to focus on assay design, experimental setup, and the factors that affect RNA isolation and reverse transcription. We'd have to save analysis of data for a completely separate webinar. Also keep in mind that much of what I discussed today is based on the MIKE guidelines. MIKE basically stands for the minimum information for publication of quantitative real-time PCR experiments. These are a group of guidelines that were put together by leading scientists in the research field. So what is the first step to designing a good assay? You really need to know your gene very well. How many transcripts are associated with that gene? Is it just a single transcript, or are there multiple transcripts? If there are multiple transcripts, you then need to answer the question, do you want to design an assay such that you detect all variants, or are you looking to design an assay that is specific? And depending on what you need to do, you need to be able to obtain a RefSeq accession number and then use the NCBI databases to identify the exon junctions, splice variants, and SNP locations. If you are doing this manually, you would have to align related sequences. So for splice-specific designs, you'd have to identify unique exons within which to design primers and probes. And for splice-common designs, you'd have to identify which were the common exons between all these variants. This is very important because you don't want to do an assay and think you're only measuring one transcript variant, whereas your design picks up all variants. So I can't stress how important it is for you to know your gene. Finally, you would need to blast primer and probe sequences to make sure that whatever you have designed is not cross-reacting with other genes within the species. In terms of primer design criteria, what you would need to do is to make sure that your primer TM values are similar within plus or minus 2 degrees. You want to make sure that both your primers find its target at the temperature that you desire. In 5' hydrolysis qPCR assays, this is normally around 60 to 62 degrees. That is where we design our primers at. In terms of length, we aim for 18 to 30 bases, but it's not really a length requirement as much as a TM requirement. It takes that much in length sometimes to achieve a TM of 60 to 62, and depending on your depending upon your GC content, that can vary the length from 18 to 30 bases to achieve a TM of 60 to 62. In terms of GC content, you want a good amount of GC in your primer, ideally around 50%. It ranges from anywhere between 35 to 65%. More importantly, we try to avoid areas that include runs of 4 or more Gs. This is because the Gs have a tendency to form a quadruplex structure, and that may affect primer binding and primer efficiency. You would then need to analyze your sequence to make sure they don't create any secondary structures, especially also self-dimers and heterodimers. IDT offers Psi tools such as oligoanalyzer, and we routinely check our primer designs for self-dimers and heterodimers using this tool. And in terms of design location, if measuring gene expression, you would like to try and design primers to span exon junctions. I'm going to touch a little bit on that in the next slide. This is not, but I also want to say that this is not necessary if you're doing a copy number estimation. For that, then you can design within an exon. However, if you're wanting to measure gene expression, then it's nice if you are able to design your primers to span exon junctions. And as before, always blast potential primer sequences and redesign if you find your primer sequence cross-reacts. So how can you use primer design to limit genomic DNA amplification? The idea being that if you design primers that are placed in different exons, these primers, if they have to amplify genomic DNA, have to amplify through an intron. This amplicon is large, and the cycling conditions for qPCR are such that it promotes and favors small amplicons. So the cycling conditions would favor amplification from your primers when the target is the cDNA and the introns are not present. The second level that you can try and incorporate is if you design your primers such that they span exons. This makes it even more difficult to amplify genomic DNA, because here your sequence now is partially hybridizing to one exon, but it doesn't pick up the other exon in the genomic DNA because it's spaced by an intron. However, in the cDNA, these two exons come together, and this primer is more efficient at amplifying the cDNA and not the genomic DNA. So this is not a requirement. This is only something if you can design a primer across a junction, that would be nice. And also the third level to keep in mind is if you're designing it across introns, the larger the intron, the better it is, because there's less likelihood of amplification of this large amplicon. Now, it doesn't mean to say if you want to design an assay with a larger amplicon, you can design it such, but just make sure that you take into account in your cycling conditions that you give your polymerase enough time to be able to extend, say, up to 500 base pairs or more. That is at which we recommend if you want to increase it. Optimally, it's smaller than that, but if you do want to increase it, you can go up to 500 base pairs. In terms of probe design criteria, this is very critical. You do want the TM of your probe to be higher than your primers, at least 6 to 10 degrees higher than your primers. This is important because you want to make sure that your probe has found its target when that polymerase takes off from those primers, because you do not want to have amplification and no probe sitting there, and as a consequence, your sensitivity would decrease, as you would not be able to detect that amplicon. So you do want to make sure that your probe is of a higher TM than your primers. As earlier, you do not want runs of consecutive Gs, and you would like GC content to be between 30 to 80% for the same reasons as your primers. Another important thing to keep in mind is you would want to avoid having a G at the 5' end of your probe. Especially with fluorescein dyes such as FAM, G base has a tendency to quench FAM. So automatically, if your probe starts with a G, your sensitivity is impacted. So we do try and avoid designing a probe with a G at the 5' end. Probe length, for most probes, the traditional probes, you do not want the probe to be longer than 30 bases, because by having it longer than 30 bases, you're separating the dye from the quencher, and the quencher is not able to be as effective. However, this is an exception for IDT's Zen double quench probes. Because of the presence of the internal Zen quencher, we can make longer probes. These Zen double quench probes are also very effective because they have very low background. And probes can be designed on either the sense or the anti-sense strand. If you're designing it on the sense strand, you want to make sure that it's closer to the forward primer, and then if you're designing it on the anti-sense strand, just make sure, if you can, to design it closer to the reverse primer. Optimal amplicon size, as I mentioned before, is anywhere ranging from 70 to 200 base pairs. If you want to design an amplicon larger than that, that's fine. You just have to make sure that your cycling conditions allow for longer extension time by the polymerase. I wouldn't recommend going anywhere much beyond 500 base pairs, but you can do it. If you're doing SYBR, then you do want your amplicon length to be slightly bigger, just because you need to make sure that you can differentiate between your primer dimers on a melt curve. And of course, you need to have primers that only amplify a single amplicon, and it's good to check it on a gel to make sure. In terms of primer location, this is a really important feature, the effect of SNPs on primer TM. This is a graph showing the amount of SNPs that have been discovered since 2002 and 2010. So in 2002, there were only 5 million SNPs that were identified. But with the advent of next-gen sequencing, you can appreciate in April 2008, we just had 15 million SNPs identified, but in 2 years, we now have 30 million SNPs. Why is this important? Well, if your primer is designed on a SNP site, it can have a significant effect on primer TM. If you see the exact match TM of this primer 62. However, if that primer was located on a SNP site, that TM will drop to 54. This can have a significant impact on primer efficiency. So any assays that were designed based on sequence information in the early 2000s is likely based on sequence information that has not been updated with the number of SNPs that have been incorporated in the database now. IDT PrimeTime pre-designed assays are designed to avoid SNPs because we are using up-to-date current SNP information. So we recently launched our IDT PrimeTime pre-designed assays. I thought I'd take a slide or two to describe the process that goes into developing these assays. There's a lot of bioinformatics involved. In fact, we need to draw on from the genomic database from NCBI as well as their RefSeq database to obtain up-to-date transcript information, and finally tie that and find all the SNPs that are associated in the SNP database. So basically, our design tool correlates the gene tables with the RefSeq information and is able to then map all the exon junctions as well as the different variants possible, and finally map the SNP sites. And we then will design primers and probes and avoid any SNP sites that are there. So all possible primers and probes are designed, avoiding the SNP sites, and then all these sequences are blasted, and any sequences that cross-react are thrown out of the database, and finally they are scored to give on secondary structure, design, location, and on even the exons they span and different criteria. So this is all that goes into designing pre-designed assays. I'm showing you the link. This is what is available at IDT. It's for available, pre-designed assays are available for human, rat, and mouse. I'm not going to go into the details of this. It's going to be discussed in an upcoming bite-sized bio webinar, but it is available on our website. In terms of instrument selection, you need to make sure that you choose your fluorescent dye based on your instrument. You have to be aware that if there is potential crosstalk between selected dyes, it's not a simple switch from just switching one dye to another. If you are testing a dye that is capable on your instrument, you may require to calibrate it. That is a very important part because I think many people underestimate the power of spectral crosstalk. Calibration is very key if you're testing a new dye. In terms of the experimental setup, if you're dealing with normal and mutant cells, you need to have multiple biological replicates for each time point that you're interested in. For each replicate, you want 2 RT preps and 1 no RT control. This no RT control is important because you want to make sure that what expression you're seeing, if you're doing gene expression, is not the fluorescent signal that you're seeing is not due to genomic DNA, but due to the cDNA. So this is a control for that. You want 3 technical replicates for each of these samples. You need no template controls for each qPCR assay tested. And another important thing to keep in mind is cycling conditions should be according to the master mix used because the master mix contains the enzyme, and the enzyme, each enzyme can be slightly modulated slightly differently. So you do want to use cycling conditions according to the master mix. And then, of course, test for your qPCR gene of interest. as well as multiple reference dyes. It's not good enough to just test one. You want to test at least 3 to 4, and the important criteria being that they be stably expressed across different experimental conditions. Moving on to the RNA and reverse transcription aspects, the quality of the template is very important because you do not want to get negative because your quality of your RNA was not the best. It is especially important for low abundant transcripts. You also need to assess if you have genomic DNA in your samples, especially if you're measuring gene expression. Inhibitors can lead to delayed CQ values or you may not even get a CQ, so you need to be aware that phenol, lipids, and starches can inhibit subsequent PCR. And the amount of co-purified salts, of course, will affect primer and probe binding affinity. This is a graph just showing the difference between having intact RNA and comparison to partially degraded RNA. You can see the shift in CQ. So qPCR quantification assumes that your samples are of equal quality. In terms of RNA sample isolation, the traditional guanidine isothiocyanate phenol.chloroform is very popular, and it's especially for troublesome tissues and small RNA isolation, but you have to do a separate DNase digestion with that. The alternative is spin columns that have on-column DNase digestion, and this is good for high throughput situations. However, you have to keep in mind that small RNA may be lost. So what method you choose is dependent on what type of RNA you're interested in, but just keep in mind that you want to use the same method for all your samples. So comparison of data obtained using RNA isolated by different methods is not advisable. In terms of quantitation, you can use UV spectrum photometry, microfluidic analytics, as well as the fluorescent dye detection. The gold standard is the microfluidic analytics because not only do you get a quantitative assessment, it also gives you assessment of the general state of the RNA sample via the RIN number. So in terms of publishing, this is a very valuable number to have, and this is the gold standard in assessing your quality of your RNA. So always use the same method of quantitation, and accurate quantification is crucial for true estimation by qPCR. Finally, reverse transcription can also be a source of error because you need to make sure to use the same amount of RNA for all your samples, as well as keep in mind that sometimes if you don't dilute your RT reaction enough, the RT reagents itself are inhibitory to PCR. As you can see, this is a 10-fold dilution, but because of the highest concentration of cDNA, that the CQ is inhibited and it comes up later, as opposed to when the cDNA gets diluted and you get the 10-fold separation of your CQ values. Priming strategy is very important. Gene-specific primers can definitely yield highest sensitivity, but that means that you can't use your cDNA for more than one type of gene assessment. In terms of oligo-DT, the advantage is that you can get the 3' end of your RNA. Hexamer priming is great if you are dealing with an RNA with high secondary structure. A good combination to use is oligo-DT and hexamer, but if you have the opportunity of having enough sample to do individual gene-specific priming, that's a good option as well. So good qPCR assays, of course, will have specific primers and probes for all the reasons we've described in the previous slides. You definitely want to make sure you have all the right controls, the no template controls, the no RT control, multiple reference genes, and a positive control. And your assay performance finally will depend on the nucleic acid quality that so your RNA isolation method is important. The choice of your reverse transcription primers, what priming strategy you use, and in your experimental setup to make sure that you have enough replicates, biological as well as technical, that will make appropriate interpretation of your data easy. So optimized assays will result in increased sensitivity, reproducibility, and specificity. These are some of the highlights of the MIKE guidelines. I'm not going to reiterate them. I've just you can look at it later when it's available for viewing. I would like to point out that for publication, sequences of the primer and probe is essential, as well as location and identity of any modification. In terms of what we have on our website, we have the pre-designed qPCR assays that are available for human, mouse, and rat. These sequences will be available upon purchase, as well as you can do live custom design for species other than human, mouse, and rat. And as I said earlier, some of these design tools are going to be discussed in an upcoming bite-sized bio webinar. And finally, much of my information for today's talk has been drawn upon from the MIKE guidelines. And thanks for your interest, and do let me know if you have any questions. Thank you.
On to sort of the more general questions. Are the CyTools that you presented available for public use, or do you have to be at IDT to use them?
No, they're available for public use. In fact, let me go to the end, and I can just show you on IDT DNA. This is our website. It's available for public use. So you have the CyTools. As I was talking, you have oligoanalyzer for your, just checking the self-dimers, heterodimers. You have the qPCR assays, the pre-designed qPCR assays, as well as the real-time custom design assays. And these are all available. This is
just publicly available.
Yeah, and I've kept a link on it on the webinar, so if they want to access it, they can. But this is the oligoanalyzer where we type in a sequence and put in your conditions, and you analyze, and you can say analyze the self-dimer, and it goes. This is going to be discussed in an upcoming, so I won't go into the details of it, but it is available for
it's okay. Okay, that's great. So it's on the IDT website. Another question I have, do you have any suggestions for dealing with repetitive sequence in your Amplicon?
In terms of design, we also offer the dual-labeled LNA probe. Sometimes it's a little tricky, so I can't give you a generalized statement. It's a little bit up to looking at the sequence, but we try and find smaller regions which are unique, and if you have enough LNA bases, we modify with LNA that increases the TM, and so then we would make a dual-label LNA probe. I would have to look at a sequence to be able to specifically be able to see what works well. You almost have to look at it in conjunction with primer, where the primer would be and where the probe would be.
Okay, so sort of
no surefire recipe, sorry.
Okay, so sort of an individual basis. Okay, the next question then, do you have any suggestions on how to go about generating a positive control or standards?
So there are two options in the sense that you could do ultramers. We at IDT, we synthesize up to 200 base pairs in length oligos. So when you know your Amplicon, you can just synthesize that region and use that as different dilutions of that as your positive control. Alternatively, you could also clone those and have genes made for you by us. We also sell mini genes, but cloning those products and making genes out of them and then using that as a positive control can also be used.
Okay. One other question then. So what is the ideal DNA purity for a good experiment, an ideal DNA RNA purity for a good qPCR experiment?
So normally they're looking at in RIN numbers, and a higher the RIN number in greater than 8 is definitely is good. You don't want your RIN numbers to be as low as 1 or 2. And even if you're looking at A260 by 280 measurements, you also want to do an A230 just to make sure how much phenol or any other organic contaminants may be coming along.
Yeah.
So that's something to keep in mind.
So sorry, were you saying a RIN number? What's a RIN number?
So if you let me go back. So Agilent and Biorad, actually, they're not IDTs, but they sell these microfluidic instruments. It's actually like a capillary electrophoresis where you separate out your sample, and it not only measures the RNA. So it has a formulation depending on the ratio of the 28S, 18S, and 5S. They can tell you in the quantitative assessment of your RNA. That's what they call the RIN number. So you have to do it either with the Agilent bioanalyzer or the Biorad Experion.
But if you don't have those, the A2A to A260 ratio should be good enough.
Yeah, 1.8 and greater. Yeah. And you also want to make sure you don't have high absorption at the A230.
Okay, and a question that's just come in, do you have any recommendations when using rapid qPCR?
No,
general recommendations.
It's a very general question in the sense I do say that you have to follow the master mix recommendation. I'm not quite sure I understood. I mean, it's a very general question, so I'm not sure.
Yeah, I think this person may be looking for general recommendations of the best. I suppose maybe the 3 best tips on how to get the best results.
No, I wouldn't go. Sorry, I just don't have any recommendations on that, and in terms of which kits would be.
Yeah. Okay. This is maybe a cost question. What is the relative cost of running a PrimeTime HP assay versus using SYBR on a per sample basis?
If you can give me that person's name, I could get them in touch with the sales representative. It is still a little more expensive than SYBR, but considering the fact that you don't have to do post-run melt analysis, and the quality of the assay is so high, and all the work that goes into designing is already taken care of, I think that little bit additional cost is worth it. But keep in mind that many times with many of our competitors, you have to buy a lot more reactions than you actually need. So with the PrimeTime Mini, what we're trying to do is we are selling it at around $75. I'm not the salesperson, so please don't hold me accountable. But the idea being that if you're in the discovery phase, you just buy as much as you need, and you have not invested that many more dollars into some gene that you may not be interested in following up on. So there is not that much initial investment. I cannot give you a per cost per reaction, but I'm sure I can work that out for you and get back to the customer if they want to give me their email address.
Okay, that's great. Okay, we'll look at another question then, which is maybe more non-cost related. Do most Proma Priber design tools look at both strands?
So I can't say for most, but our design tool will be looking at both strands.
Okay.
For our PrimeTime pre-design qPCR assays, we would look at both strands. But I do not know. I cannot say for the other tools.
Okay. When would you recommend using a double quench probe?
Actually, if I have a customer who's just using FAM Iowa Black FQ, especially it's FAM. FAM is the fluorescent dye. I would recommend them using the double quench probe, because in head-to-head comparison, the double quench probe has so much lower background than any other dye quencher combination tested, that to me that should be the first choice if you're using FAM, to use a FAMs and double quench probe.
Okay, somebody's asking a question actually on the dye and instrument capability. I mean, I saw that you put the slide up, but someone's wanting to know if that data is available anywhere else, as they want to know if there's... They want to
On
know
the
if their
IDT
SYBR is
website,
Yep.
we have in our support section for our PrimeTime qPCR products something called a qPCR application guide. We have a lot of supporting documents, and you can even ask the sales rep for the qPCR application guide. This is available within that guide. So we basically have taken a list of all the different instruments and charted out which ones are instrument capable dyes but may require calibration. Those are the open circles. And then the ones that the instrument is just not capable of supporting, those are the crosses. So we have that charted out, and it's in the qPCR application but guide, and it is available on, let me see if I can link out. Let me show exactly where.
But anyone should just be able to find this on your website?
Yes, under the PrimeTime pre-designed qPCR assays, under our support section, and it's here. Okay.
Oh, yeah. Yeah, okay.
Sorry.
This is where you go. No, that's fine.
It's supposed to be right here.
Yeah, I mean, what we'll be able to do is this webinar, it will be available as a video, so if people are wanting to just scroll through and have a look at the end, they should be able to see the web link there. So that's great that you've got that there. Okay, so coming back to a question. How do I decide what combination of fluorophore dyes I need for multiplexing?
So actually that brings us back to this slide again. So it really depends on your instrument. Some instruments are better for multiplexing just because they have a broader range of dyes that are compatible with that instrument. So when a customer calls in, normally we really make use of this chart. So I always have FAM. FAM is a very reliable dye, and then spectrally I try and see what is calibrated on the instrument. And so suppose it's BioRad, you can do a FAM hex, if you want even more than that, then you would go across this chart and see, okay, it's calibrated for this instrument. I can use FAM, hex, and Texas red. So this definitely helps in terms of identifying what dyes to use for multiplexing. You want to keep them spectrally apart to minimize crosstalk. That's really very important.
So actually this next question may be related to that. What should I use as a quencher on the instrument when using a dark quencher?
So that's something that comes because I guess every instrument they have different ways of naming it. ABI sometimes even calls it MGB. Anything that says detector none or MGB, you can use that for a dark quencher setting. You do not want to set it on a camera setting. Anything that says for the detector setting either none or MGB, you can use that when you're using dark quenchers.
Okay, so you've said MGB. What's MGB? I'm not sure of that one.
So MGB is a minor groove binder. It's licensed by ABI and sold exclusively through ABI. It helps to raise the TM of probes, but it's not needed in terms of functionality of the assay. You can design dual label hydrolysis probes, and as long as you design them longer to account for sufficient TM for gene expression, it's not needed. But that is something that we come across also quite often because people tend to pick sequences directly from a paper but are not cognizant of the fact that these may have been MGB probes and they're shorter in sequence, and their TM may not be right for it to optimally function in an assay. So you need to keep that in mind when you're just taking someone's paper and trying to recreate it, that it not be an MGB probe sequence. And we can work with it in the sense that we can definitely lengthen and find the best probe. We don't just need to lengthen the probe. We can find the best probe and use that for the assay.
Okay, so don't just copy the paper.
just keep in mind the TM. If their sequence of their probe is around 15 Mars, that's likely it's an MGB probe because that TM is likely going to be much lower than 65. And so that's a way to know. The length, if it's less than 15, definitely 15 to 18, maybe.
So there are some key things there you can maybe look at to know whether
Yes.
it's MGB. Okay, so maybe we have time for just one more question. So is there any reason to design probes to one strand versus the other, sense versus antisense? I guess that you would want to consider both strands to give you the probe with the best specs.
Yes, you don't need to favor one strand over the other strand. You just want to make sure that if you're designing on the sense strand, that your probe be close to your forward primer, because as soon as your polymerase takes off, you would, as much as possible, you would like it to be closer to the primer, and it immediately as it extends off, it cleaves off the probe, you don't lose sensitivity. So and the same token, if you're designing it on the reverse strand, make sure that you got the right sequence in terms of the orientation, as well as if it's closer to the reverse primer, because that probe is going to be cleaved off by extension of the reverse primer. So you don't want your probe sequence and your primer sequence to overlap, but they can be designed on either strand.
Okay, so it doesn't matter which strand you use.
Yeah,
okay.
The more important thing is TM and making sure it's the right TM.
So you're just matching up the TMs, those parameters you discussed earlier first. Okay, well, I actually think that in terms of time and the questions, that brings us to the end of the web seminar.